Our next presentation is Kiji Therapeutics, and I'd like to welcome Miguel Forte, CEO, to the stage.
Good afternoon, everybody. I'll stick to my 15 minutes and get you to lunch. So, what is Kiji Therapeutics? We address the problem of unmet medical need, particularly Treg unmet medical need in autoimmune diseases, through the use of a cell therapy. The solution is very simple. Use MSCs from iPSC source and engineer them for potency purpose, so really delivering a enhanced stimuli that will drive Treg function. It's a proprietary platform for a bicistronic gene that we use currently with a lenti transduction, and we've been transducing CXCR4 IL-10, and I'll come back into that. We've been established in March 2023, and we aim to be in the clinic in the middle of next year. A quick differentiating purpose: we bring a platform that will anchor on existing data, existing experience, but will take it to the next level in terms of benefit. We expect, and we would be delighted to see, the approval of Mesoblast at PDUFA data in 7th of January next year for their MSC-based approach for GVHD. With an approach that we bring, we will be able to increase the therapeutic delivery by well over 50-70% by being able to really have a focused local deliver of IL-10 with a consequent anti-inflammatory stimuli. So we see really a very differentiated, significant opportunity coming our way as we go into the clinic. We have an experienced team. Both myself and Tony Teng, our CSO, are around and more than willing to have discussions and chats. We're quite busy, but definitely keen to engage. The pipeline that we have is the first product going to GVHD. It is in late stage of IND enabling studies. We will have the engineering runs early next year and the subsequent IND application, aiming to be in the clinic, and I'll come back to that, aiming to be in the clinic by middle of next year. That first indication will be a proof of concept that we're able to enhance the use of the MSCs, save cells, but enhance their potency by the gene engineering, and opening the door to Kiji 02 and 03, where we'll be able to expand to other indications, namely IBD and psoriasis, as well as undisclosed indications with additional genes. And so it will be crucial to achieve that clinical data, because it's not only delivering value to unmet medical need in GVHD, but will establish the platform to much higher ambitions like IBD and psoriasis. And you will know, all of you, don't need to elaborate on the market opportunity in those indications. But the key element is going to be delivering it to a targeted patient population, and I'll come back to that point in a minute. The first product is thus anchored on the effect of IL-10. The CXCR4 IL-10 that we transduce makes the cells go to the site of inflammation, driven by the CXCR4 connection to the STF1, where they offload large quantities of IL-10. We have all the elements of evidence of that mechanism of action that is here shown here. We have the inflammation. The inflammation corresponds with large amounts of STF1 that drive the cells to the site of the inflammation where IL-10 is produced. All of those elements we have documented in in vivo and in vitro in terms of the different steps of mechanism of action. We do have in vitro evidence of migration, of increased immunomodulation by higher levels of IL-10. We have in vivo animal models showing an increased benefit over unmodified MSCs in GVHD, IBD, and the skin model. And we have the evidence in terms of the biodistribution, the specific IL-10 production, and the cellular changes that we induce. We indeed induce increased Tregs, Bregs, and in the IBD model, we actually do a macrophage switch, providing an opportunity for long-term benefit through innate immune system resetting. Just a couple of snippets of information from our data. In vitro, you can see here the migration being driven by the CXCR4 and not by other modification. And conversely, we see an increased immunomodulation with the IL-10, with or without CXCR4. So really building blocks of the effect. In the in vivo model, we see a clear differential benefit, and I'll take you through the data. As you can see in the untreated animals, the benefit that you achieve with unmodified MSCs, which is well described, recognized, and already in the clinic and in anticipated approvals, as I mentioned to you before. But then over and above that, we achieve an additional benefit, an incremental benefit by the transduction of the MSCs with CXCR4 IL-10. I mean, the idea is pretty simple. We know that the cells have their mechanism of action driven not only, but primarily by these two factors, and enhancing them, we're really just giving an additional potency to that benefit. But we can go beyond that. If we have the IL-10 associated with the mechanism of action and associated with the clinical benefit, will be the anchor for a potency assay that enables us to have an easier, well-established CQA for the product. If on top of that we then use an iPSC as it is our intention, we will then have solved the two main issues that have been resulting in insufficient therapeutic benefit with MSCs, which is the CMC element solved by the iPSC and the potency element solved by the gene engineering. That's in one sentence the message. But the mechanism behind that message in terms of Treg induction is particularly relevant because we should target, and we are targeting, Treg-dependent autoimmune diseases. And having elucidated in the context of an IBD model the macrophage benefit, we'll be targeting patients with a macrophage-dependent IBD phenotype. There's a switch on the cytokines. There is the evidence of the biodistribution where the transformed cells go to the site of the tissues in inflammation and produce IL-10, really elucidating the mechanism that I've highlighted to you. If we go to the IBD, we in a DSS-induced model, we have again showed the increase in benefit, but what we've shown is that that benefit is sustained long-term after a re-challenge without a second treatment, apparently translating an induction of a switch in the innate immune system, ensuring that long-term benefit that everybody tries to pursue in the context of treating IBD. Trying to elucidate that mechanism, we looked at the different cell type modifications in those animals, where we have again confirmed the Treg increase, but we've also documented, interestingly enough, a switch on the macrophages from a pro-inflammatory to an anti-inflammatory profile, together with myeloid-derived suppressor cells. As you may know, recently patients with a certain genetic marker have been associated with the macrophage phenotype, giving us a clear opportunity to start the development by approaching those patients, giving us a higher probability of success based on the mechanism, being able to not only have a short-term benefit, probably Treg dependent, but also a long-term sustained benefit, potentially macrophage dependent. This is the key to that benefit. So in essence, we're targeting GVHD, IBD, and skin in this process, with the manufacturing is relatively simple. It's collection, expansion, transduction, and formulation, cryopreserved, off-the-shelf, allogeneic approach, clearly to be clinically compatible and easy to use. We'll switch this to an iPSC sourcing, still based in the first approach on a lenti transduction, but we've done gene editing, nucleotide and base editing of the cells, also to do additional modifications, namely to consider long-term permanence and increased tolerability. The clinical study that we're aiming to do in Spain, the sites are selected, investigators are selected, the protocol is defined. As I said, we're in late stage of IND-enabling studies. The intention is to start second half of next year and deliver the data within 12 months from that. A small number of patients, open label, really to give us the opportunity to document the benefit and the safety in late stage doubly refractory patients, 25% about of the patients in GVHD, where the unmet medical need still remains. So very quickly, in terms of the strategy of the company has been created. We had an initial seed that has been complemented with grants. We are currently on a process of additional funding already secured for short term and on the road for an additional funding that we aim on short term 10 and long term 60 that would enable us to deliver in terms of clinical data for GVHD, initial clinical data going into the clinic in IBD and psoriasis, and really expand the platform to an iPSC-based gene-edited MSC platform. There you go. Happy to have any questions or engage on discussions out there. Keen to be in the meeting and really excited on the opportunity we're bringing. Streamline the presentation, but I think you got the gist of it. Thank you very much.